Well, let us discuss the last part of the immune system, the third line of defense, the specific immunity. All right, so we're going to start off by discussing T lymphocytes. T lymphocytes are so called because they become immunocompetent inside of the thymus. So we have that tie-in between the T in the thymus and T lymphocytes. And this process of immunocompetence happens in about the first 2 years of life. After that point, the thymus is kind of degraded inside of the body. And that process is pretty involved, which we will sort of touch on a little bit later when we talk about diseases. All right, so when T lymphocytes are going to be activated, they have to be stimulated by self, non-self complexes. The self part of this being antigens or proteins bound to the membranes of self cells, cells that were produced in your own body, in your own body's cells. Non-self would be considered pathogenic antigens, proteins that are bound to the surface of some kind of an other enemy, bacterium, virus, anything like that. So in the last video, we discussed antigen-presenting cells, and we sort of alluded to the idea of the self-non-self complex. So the self protein here is going to be attached to the side of the antigen-presenting cell, and it's going to be, in this case, presenting a small part of some kind of a pathogen that it has been consuming. Additionally, though, we can have self-non-self complexes that are produced by any kind of infected or affected cell within your body. So when you have an infected cell, it's going to be rife with bacterial parts or virus parts, something going on inside of there, and therefore their cytoplasm is going to have a sampling process, and that sampling then is going to bring particles of that, little parts of that pathogen out and attach to the side of that self protein. Now, the story with antigen-presenting cells is a little bit different because remember that an antigen-presenting cell, it's a phagocyte that was able to sequester whatever particle, whatever virus or bacterium or pathogen, and sequester it inside of a membrane so that the cell is not actually infected by it, but it can be quite purposeful about bringing that phagolysosome over to the edge and presenting in just the same way, really, but with different consequences, of course. The antigen-presenting cell is just fine. The infected cell, not so much. But the sampling is still the same. It's able to bring a small particle of that pathogen to the edge of the cell, basically, and then there will be a self protein that then presents that non-self pathogenic part. Well, let's talk about what those self proteins actually are. These are major histocompatibility complexes. An antigen-presenting cell will have an MHC class 2, a major histocompatibility complex class 2. And the infected cell, and in fact, every cell of your body has MHC class 1, major histocompatibility complex of the type of 1. And so when we understand then that the interaction here comes from the white blood cells, it turns out that particular types of white blood cells are attracted to different types of MHCs. So the CD4 cells will be attracted to MHC type 2, and which would be on the antigen-presenting cells again. And the CD8 cells would be attracted to MHC class 1, which would be on any cell of the body as long as there's something going on with it, maybe some kind of damage, cancer, infection, something like that. Now, additionally, we do have to draw on there a co-stimulatory factor, a small protein that would stick out of the side of either an antigen-presenting cell or an infected cell, and this would basically confirm that yes, not only is this particular cell presenting a problem, but there are other things going on. There's an inflammatory response going on. There's many cells involved, something like that. The co-stimulatory factor is a protein that sticks out of the side of the cell and allows for just that extra kind of oomph to really allow the white blood cell to understand, yes, this is a problem. So let's be very clear. We are looking at an antigen challenge here. The antigen challenge involves a self-non-self complex. In the case of the CD4 cell, it's going to be a class 2 MHC. And additionally, there has to be a co-stimulatory factor to get this thing actually activated. And it takes about 7 to 10 days, and this cell does begin to clone as long as all those characteristics are in place. The same here, just to be redundant and be very, very clear, the antigen challenge works exactly the same way with a CD8 cell and infected cells, which is that you have to have a self-non-self complex. In this case, the self part would be an MHC class 1, And you also, again, have to have that co-stimulation, some kind of an assent that says, yes, not only is it just me, I'm not the only problem, but we also have all these other issues too. And if that is the place, then we start to clone these guys. Again, 7 to 10 days to clone them properly. So let's take a look at the CD4 cell. Again, we're looking at a T lymphocyte that is called a CD4 because it has CD4 glycoproteins attached to its surface. When it clones, it's going to first of all create memory CD4 cells. And no surprise, when the CD8 cells, again called CD8 because they have CD8 glycoproteins sticking out of their surface, when the CD8 cells begin to clone, first of all, they make memory CD8 cells. And these memory cells are stored away for later infections by exactly the same pathogen in future, where they will be activated much more quickly and mount a much more specific and powerful response against it. So what else? The CD4 cells will be able to clone themselves into T sub H cells or helper T cells. Helper T cells are called the managers of the immune response, and with good reason. Although they themselves do not actually consume anything or directly attack anything, they basically enhance everything that goes on basically in the immune system. So first of all, they're able to activate your T sub C cells, which we will talk about in a moment, cytotoxic T cells. Secondly, helper T cells can actually release cytokines, which I think that cytokines do a number of things, but you should be remembering that they are responsible for the inflammatory response. So helper T cells help with inflammation. And third, helper T cells have to help to activate B lymphocytes or B cells, which in turn you should be thinking about antibody release. We'll be discussing those in a moment. So the helper T cells do a number of things that kind of command the signals between other elements of the immune system. Now CD8 cells are able to become what we call T sub C cells or cytotoxic T cells, and I'm just going to put in parentheses killer T cells. This is what they used to always be called. So killer T cells are able to just absolutely take out any kind of an infected cell to make sure that it is no longer spreading harm. Let's take a look at how this looks at with a cancer cell. So the cancer cell tends to be quite large. You'll notice, of course, that T lymphocytes, you may recall, lymphocytes in general are relatively small cells. But as small as they are, they can actually take out this enormous cancer cell by injecting stuff into it. They're going to inject perforins that perforate the membrane. They're going to inject granzymes, lymphotoxins, and what these things do is they basically degrade the cellular contents of the cancer cell or any infected cell of the body or old weakened cell, anything like that, and therefore the cell undergoes apoptosis or cell death. So you can see that this is quite an important role, kind of a dangerous role too. You want to get it right every time, so that programming, making sure that the T lymphocyte is acting right is quite important. So this gets us into disorders. What happens when T lymphocytes, for example, cytotoxic T cells, are not programmed right? We might have an autoimmune disorder. So an autoimmune disorder is a situation where your self-tolerance breaks down. You basically start to not recognize your own self as self, and for whatever reason, the immune system starts to attack self-tissue. So the first and probably most common way that we experience this is through erroneous lymphocyte programming in the first place. This would be very likely due to a genetic issue, which could infer that oftentimes autoimmune disorders are actually genetic. So what's happening here is that the original, for example, when the T lymphocytes were inside of the thymus and becoming immunocompetent, unfortunately something went wrong and you started with T lymphocytes that were not programmed right. I don't know really how else to say it. They were somehow, I mean, there's such a plethora of autoimmune disorders that it's hard to just conceptualize them all by a few words here, but the basic idea is it's possible that you might have let through some T lymphocytes that were supposed to be undergone, apoptosis themselves. Only about 2% of T lymphocytes actually become T lymphocytes. Most of them they're meant to be, they seem to be too overreactive, and so they're basically taken out in that process. But sometimes some slip through that aren't right. They don't act right. And therefore, this is one of the ways that you could have an autoimmune disorder. Again, it's broad strokes of a brush. The reality is we really don't know enough about autoimmune disorders. That's still true to this day. We're getting better at diagnosing and treating, but there's still a lot of mysteries there, to be honest. Okay, the second way that autoimmune disorders can occur, again, just broadly speaking, is that you could have no new proteins that appear in cells, and therefore the body recognizes these as pathogenic, but they are, of course, self. This might actually come about because of this might come about because of mutations that would randomly form new proteins. So they were manufactured by your body, but your body recognizes them as a pathogen. And then lastly, foreign antigens may actually resemble self-antigens, and therefore, as the body attacks this foreign antigen and becomes kind of sensitive to it, then it starts to recognize some of your own body's antigens as the enemy as well. The most famous or common way that we discuss this is through rheumatic fever. When a person undergoes rheumatic fever, the body is vigorously fighting off these bacteria that cause this. And unfortunately, those bacteria are very similar to cells inside of the heart. So sometimes when a person has rheumatic fever, the body fights it off. Then the cells that fought off that bacterium will begin to turn against cells of the heart. And people who have rheumatic fever oftentimes down the line will end up with a weakened heart because it's an autoimmune condition where these T lymphocytes or these immune cells are attacking cells of the heart. All right, a little faster topic here, immunodeficiency. It's actually pretty straightforward, although there's different types of it. Basically, immunodeficiency is a problem where you have suboptimal performance of the immune system. So there's two basic ways that this could occur. The most common way of genetic or hereditary issue would be SCID, severe combined immunodeficiency, where basically either the T lymphocytes or the B lymphocytes usually are just not manufactured at all in the first place. This is a developmental problem. It can be cured with a bone marrow transplant, sometimes in utero, amazingly. And then the most famous type of immunodeficiency that you've definitely heard of is AIDS, which is an acquired version of immunodeficiency. So SCID is genetic, you're born with it. AIDS is acquired, and of course AIDS is acquired through acquired immunodeficiency syndrome. HIV, the HIV virus would be the cause here. Now the HIV virus is going to be attacking specifically the CD4 cell. The reason why the HIV virus is so dangerous is because it takes out, remember, the manager of the immune system. And anybody who has suffered from AIDS or has witnessed anybody suffering from AIDS understands the importance of those helper T cells. Although they themselves do not specifically, consume anything or attack anything or produce antibodies, for example, they have a pivotal role in enhancing every other aspect of the immune system. And so you can see that a person who starts to have their helper T cells degrade over time because they have AIDS, people don't really die of AIDS, but they do die of secondary infections that the body was unable to fight off, pneumonia or tuberculosis, for example. Well, we've gotten a lot better at treating AIDS, of course, but usually the cocktail treatments will include some kind of cytokines. Cytokines would include families such as the interleukin families, and so oftentimes, if you know anything about treatment for AIDS, it's going to include interleukins or some kind of cytokines. So that kind of points to the fact that you're trying to augment some of the roles that the helper T cells were. Remember that helper T cells secrete cytokines, and so or produce cytokines and release them, and so that is something that they try to do to help patients who have AIDS. Well, let's completely change topics then, and let's talk about the B lymphocyte story. The B lymphocyte story should always be combined with the idea of antibodies. Please remember that. And this is what we call humoral immunity. Remember the body's humors, interstitial fluid, blood, lymph. You're going to find lots of antibodies, especially when you're fighting off an infection. You're going to find lots of antibodies in those bodies' humors. So it's called humoral immunity. So what we're going to do is take a peek inside of one of our germinal centers. You may recall that germinal centers are chock full of B lymphocytes. Germinal centers are those little structures, the centers of lymphoid follicles. And so whenever you find most lymphatic tissue, you'll find lymphoid follicles. Inside of them, you'll find germinal centers. And inside of the germinal centers, you will find B lymphocytes. But we're going to go a little further, and we're going to say that most of these B lymphocytes are going to be called naive, which means that they have not yet been challenged and they haven't developed a sharp ability to fight anything off. You need that challenge to actually fight something off. So each of these three B lymphocytes, I'm going to draw in different colors of basically membrane-bound antibodies, these proteins sticking out of the surface. You notice that each of the B lymphocytes has exactly the same, like all of the antibodies on its surface are exactly the same. And so I just colored the three of them with different color antibodies to give you the understanding that each of the B cells are different from each other, but all the antibodies on one surface are the same. So what we're going to say is that this center one here, which the antibodies are colored in purple, it's going to experience an antigen challenge, which I'm drawing now, and therefore it is going to be cloning. And in this case, what happens is it's a lot more of a simple story with the B lymphocytes than it is with the T lymphocytes. All the B lymphocytes need is actually a direct challenge by the pathogen. The pathogen will directly attach to those externally membrane-bound antibodies. It takes about 3 to 5 days to begin to clone. Much simpler than the T lymphocyte story. It doesn't take much to activate a B lymphocyte, just the presence of the pathogen itself. You don't need a self-non-self complex or major histocompatibility complex or a co-stimulatory factor. It's literally just the presence of the pathogen. Right, so when this B lymphocyte clones, of course, it's going to make some memory B cells that are going to be tucked away and try to recognize the same pathogen in the future. But it's also going to clone into plasma cells. Plasma cells are big old cells, and we go ahead and draw the cellular content to remind you that these guys are going to be manufacturing massive amounts of protein, so those organelles are important for that. So this is a plasma cell, one of our larger cells. A plasma cell is able to manufacture massive amounts of free-floating, loose, now released into the humors, antibodies, these little Y-shaped proteins. And there's a lot of them. The plasma cells will release about 2,000 per second over the course of 4 to 5 days. So a massive number of antibodies as soon as those plasma cells are cloned. So let's talk about what antibodies actually do. Antibodies are basically free-floating antigen receptors. They're tiny little proteins that are able to search out and find and hunt down randomly, but still there's so many of them. They start to hunt down any of these pathogenic antigens that they find. Probably the best role that we know of for the antibodies that's most understandable is agglutination, where these Y-shaped structures will bind together lots of the different, clump together and then release harmlessly lots of different pathogens. Additionally, antibodies are capable of tagging pathogens for destruction by other structures. You know, antibodies themselves are tiny and cannot opsonize anything or consume anything, but they can make pathogens more readily opsonizable. Remember what opsonization is. It's where the complement proteins add like a sugar coating on. And in turn, also, this makes the pathogens more capable of being phagocytized by those phagocytes, neutrophils, and dendritic cells, and macrophages, and so on. And lastly, antibodies are actually capable of neutralizing toxins that might be secreted by the virus or pathogen or the bacterium. Right, so let's go ahead and look at a timeline here of how many antibodies are produced and over what timeline. So on the x-axis, we're going to write time in days. And I'm not going to put numbers on here. I think it can differ quite a bit depending on a number of factors. But we're going to go ahead and put our first encounter with that pathogen at about day 0. And about 3 to 5 days later, you're going to start to have a significant number of antibodies. On the y-axis, we have labeled this antibody titer, which means antibody concentration. If you took a drop of the person's blood plasma and measured the number of antibodies, this is what we'd be looking at. So the first tiny little bump is going to be a primary response. But with the second encounter, we have a secondary response, which you'll notice is much larger, very immediate, very long, and it basically lasts a long time. The primary response might only last about a week, but the secondary response can last a month or more. So it turns out the secondary response is mediated by those memory B cells, which we discussed up above just briefly, but don't forget about them. They're super important. But the primary response is mediated by naive B lymphocytes. And naive B lymphocytes need some time to kind of wake up, get to be to the level where they have sharp recognition of that particular pathogen. Here comes the SARS-CoV-2 virus, the one that caused COVID-19. We're still dealing with a global pandemic. And those B lymphocytes are able to sharply recognize that particular pathogen by recognizing some of the antigenic determinants on its surface, but only after some time. Whereas memory B cells already have all of that advantage. They've already recognized it, they already have a sharp recognition, a sharp ability, and therefore when they start to clone, first of all, they clone very quickly, but they already have exactly what they need in place to immediately create more plasma cells, even more this time, and to manufacture tons and tons of antibodies. Well, this is the advantage of a vaccine. The vaccine basically replicates a first encounter with the pathogen without actually having to get sick. So vaccines are usually dead or weakened pathogens or little bits of those pathogenic membranes, the outer portion of that structure, and therefore the body starts to recognize it and kind of mount a defense against it from the naive B lymphocytes, starting to manufacture memory B cells and plasma cells and so on. So that if and when the person actually encounters that in the future, they've already had what seems to be their primary response. And so their secondary response, again, very fast, very long, very hardy, and they mount a powerful response against it. Remember that you don't actually have to get sick to have your immune system recognize something as an enemy. You just have to expose your immune system to the pathogenic antigenic determinants, like a little portion of those proteins sticking out of its membrane. So I think this is a good time to kind of bring up passive humoral immunity, where you can actually donate antibodies to another person or from an animal to a human, something like that. So passive humoral immunity is different from a vaccine. A vaccine is actually going to cause a primary response. Passive humoral immunity is simply donated antibodies. It doesn't cause any kind of a reaction of the immune system at all. It basically just boosts your immunity for a temporary period, maybe about 3 weeks, until the proteins begin to degrade. There's a few ways that we can donate antibodies. We can literally inject them. For example, you can take the blood plasma of somebody who's already recovered from an illness, spin down their blood, take their blood plasma, find those antibodies, and literally just inject a sick person with the cured person's blood plasma. And as long as it's been just a few weeks since the person fought off that illness, they probably have tons and tons of antibodies in their blood plasma, and therefore this injection will help to boost the sick person's immune system. And then additionally, antibodies can be passed through breast milk. For example, from mother to baby. Another way that we see antibodies would be through an injection after you get a snake bite. Anti-venom is actually antibodies. Remember, one of the roles of antibodies is to block toxins. So this is a good example of that. So passive humoral immunity, it's just simply proteins donated, proteins given to a person who needs more antibodies and for whatever reason is not manufacturing them themselves.